From Microbe TV, this is Q&A with A&V. I'm Vincent Racaniello, and joining me tonight from Chevy Chase, Maryland, Amy Rosenfeld. Hello, Amy.
Hello, Vincent. How are you tonight?
I'm very well. And yourself? Is everything good?
Yeah, it's
Is
fine.
it hot down there?
It's
Is it hot?
It's very hot. It's very humid. I water my plant, so it's good.
It was very hot today in New York, and then around 2 p.m., just as we started TWIM, these thunderstorms rolled through, boom, 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 and it dropped many degrees, and then it went right back up again. And when I left tonight, there was no drops of anything on the sidewalk. It was quite interesting. Hello, everybody. Hello, intrepid virus hunters. Should I call everyone virus hunters?
What virus are they hunting?
Well, they're hunting information on viruses. They come here for that. And so welcome all of you. And thank you for our moderators. So far, I have seen Frank here tonight, and I have seen Steph and Vanity. I know that Tom won't be here. He's going to a play. I just saw Tom in the last few days. I'm going to show you a picture of us together. So thank you all for coming and moderating.
Wait, what happened? So you said Tom and Vanity, Steph, and Frank, but where's Les?
I don't see Les. I didn't see him. I haven't seen his, and usually he's chatty, so he's not there. And I want to thank them and everyone for coming on an odd night, because last night I had just arrived here at 8 p.m. from the airport, so no way I could have done it, because I was in Madison for a couple of days. And I just want to show you a little bit of what went on in Madison. So one of the things is that I was awarded the Wolfgang Yaklich Medal for lifetime achievement, in virology. So there's the president, Felicia Goodrum.
Yeah, from Arizona, right?
Yes, is from Arizona, giving me the award. And then, by the way, these things around our necks, those are called nerd flags, okay?
Nerd flags? Okay.
Doug Lyles, he coined that term, nerd flags, because that's what they are. You walk into a bar with one of those on, and you say, what are you wearing your nerd flag for? Anyway, here's the box. I wanted to show you folks, my beloved live stream watchers. There it is. And Lorena Passarelli had the box made. She's made of wood, And then inside is the medal, which has a likeness of Bill Yaklich. Now, who was Bill Yaklich? He was one of the founders of ASV, did a lot of pox virus work, really quite a giant in virology. He died a couple of years ago, so they established this award in his honor. And it's the second time it's been given. And then, of course, on the other side, they have my name engraved there, and it says, the American Society for Virology honors Vincent Racaniello in recognition of significant and excellent service contributions to the society, and I might say to virology in general. So this is going to the incubator, okay, Amy?
Yeah.
Amy's very happy that I got this prize, aren't you?
I am.
So then,
sure.
I you saw the picture there, and then bingo.
Wait, I was going to say, where's the Wimbledon pose?
I thought Wimbledon. I didn't kiss it, though. I should have kissed it. I look mad. Look at that. I look like a crazy man. Holy cow.
I don't think you look mad. I think you just are like screaming and exciting.
I had a blast at this meeting. Let me show you a couple of pictures. Here is Tom, our moderator, Tom Steinberg. And this is where we used to have our meals here. And Tom was very excited to be there. We talked a lot, and
he even bought a T-shirt. Look, it says
he bought a T-shirt.
2022.
Yeah. So thanks for coming. Cool. His mother lives in Madison. I think she's 90-something. So he goes to see her now and then. Cool. Tom, it was a lot of fun. Okay, so that's Tom. And who do we have now? Oh, Doreen and her husband came to visit, and I see Doreen is here tonight, and they took me to lunch at the Great Dane, which is a lovely pub not too far from the convention center. So thank you, Doreen and her husband. He had a TWIV shirt on, the miasma T-shirt, which is very cool.
Cool.
Okay, and then what else we have? We have the TWIV crew, and then we have Steph from Immune and Nels from TWIVO, who were there for the meeting. So Kathy and Rich, myself, and Brianne, after we'd done the show. And it's going to drop this weekend. Really good, two really good guests. Here's
Eva Harris.
Gosh, I can't.
Eva Harris.
Who's at Berkeley. And always a great smile. And behind us, they asked people to draw their favorite virus and put some facts about them. And they give you blank pages. And there's two bulletin boards worth of those things. This is really good. That was really fun.
So did you put up polio?
I didn't put up polio. I was too busy getting pictures taken with fans. And so they had the Virolympics, a bunch of contests where you would get points, and then at the end you get prizes. So this was one of the stations called In Vivo Imaging, where you had to find these people and take their picture with you and return to get your points. So I'm very proud to have been on this. And a lot of people came up to get that. And what else do we have here? We already showed that. I guess that's it. Yeah. We showed them and them. Yeah, that's it. So I won't give too many more because then people get mad that I'm not taking too long to get going. But
I don't think so. That's silly.
There are more pictures, but that's enough for today. It was a great meeting. Great science. The TWIV was awesome. I met hundreds and hundreds of young people who love what we do, and I really appreciate that. And a lot of the science, I'm going to try and bring in the people who talked about it here on TWIV and over the next few months because it's really exciting and you should know about it.
Well, right, even ones who weren't there but whose spouses were there, like Emily. You're going to have Emily talk about H5 again, right?
Yeah, so if you remember, we had Emily from Colorado Department of Health. They had the first alpha variant in the U.S., and Amy knows her husband, Brian, right?
Yeah.
And now they have first H5N1 from a human in the U.S., and so maybe she can come on and talk about that, right? Are you going to broker another visit?
I told him. Yeah, I did. I wrote him and said we're going to have Emily come back after after you have Kieran for the polio.
Yep. So. All right, let's see what people are thinking about here. Of course, polio comes up. Momo says, I bet you guys are all excited. I wouldn't say, I don't want people to get polio. That's not the right word, is it, Amy?
Not for me, no.
Les is here. Thank you, Les, for coming.
Right.
Hope.
No, I'm not excited. I'm... Let's see. I am hoping that this is an eye-opening event for changing some policies that have not been well thought out by because they aren't really proposed by scientists. And people didn't care when it was happening in Africa, and maybe we cared a little bit more about it in London. And we didn't really care, or it wasn't really public about it in the Ukraine and in Israel. But now that it's in the United States, it's a whole different ballgame politically. And actually, I just had this conversation with my guy this morning, and we were discussing it. And he said to me, no, it can't happen here. And I said, I bet you if it does, it's happening in Westchester. And 2 hours later, he emailed me.
So why don't you explain why this case appeared in Westchester, of all places?
Because it has a very, very low vaccination rate. It's like 10%. And you need to have at least 80% vaccine coverage to prevent any vaccine-associated polio virus transmission. And so this shows that it's there, and it shows that he was unvaccinated, and it shows that it's spreading.
So this was sequenced, and it was found to be vaccine-derived type 2
2.
poliovirus, right? Which, and Amy and I have said on this live stream multiple times
And
that
I got a lot of shit for it.
we should be sequencing, we should be surveying the sewage for poliovirus, because we said we know it's here. We use IPV, an activated polio vaccine. So our intestines are fully susceptible to any vaccine-derived strains that might come from countries that still use oral poliovirus vaccine. And we know in the UK last month, right, they found RNA sequences in the sewer. But this is a case, which means there are probably many other people infected, right, Amy? Because a case is 1 in 100, 1 in 200, right?
It's 1 in 1,000. If you're unvaccinated, it's probably 1 in 10,000 if you're surrounded by a vaccinated population. And the truth is why we and Canada have switched to IPV. OPV is still used in Mexico and South America and in Central America.
So parenthetically, so what's the solution? Either switch to IPV or make an OPV strain that doesn't revert.
Have to produce. Well, it does revert, but it's less frequent. So the point mutation in the iris, which is the neurovirulent determinant, is less frequently to revert in Raoul and Andy's NPOV2. But that only covers NPOV2. And so they're making NPOV1 and NPOV3. We're testing it.
Will there be more cases? So this case presented clinically a month ago, right?
And it was actually misdiagnosed.
What did they diagnose it as?
So I heard about it actually a month ago. The person who told me about it reminded me that. He said, oh, there's a case of AFM. And then he came back and said, oh, it's not a case of AFM. And then it was a case of, then it was polio.
Well, what did he think? It was entero-associated AFM, non-polio?
No, they thought it may have been just meningitis or something or other, not AFM. But so first they said it was AFM, then they said, no, it's not AFM. And then they confirmed it being polio. But the real problem is now AFM is a clinical diagnosis.
Right.
It's a clinical disease. It's not, so that compounds it.
So
dramatically.
Amy, do you think there will be more cases?
For sure.
In the U.S., in the same area, you think?
Yeah.
But the thing is now they're encouraging people to get vaccinated. So
they're opening they're opening 2 vaccine clinics Friday and Saturday.
That was fast.
Yeah, that's what the guy, Koishi, who sent me the article this afternoon, because we had just discussed it this morning.
So that's your boss at FDA, right?
Yeah, that's the vaccine. That's the vaccine guru.
Okay.
You know,
I thought I was the vaccine guru.
He's the polio vaccine guru. I'm sorry, sweetheart. But his father worked with Sabin. Did your daddy work with Sabin?
No, not even close. He worked near William Carlos
so
Williams.
Well, but your dad did not work with Sabin.
Okay.
His dad and his brother and
I know.
him. Yeah, it's a little different.
Okay. Anyway, go ahead.
So, what were we saying?
More cases.
Yeah. So then I told him, like 20 minutes later, I said, "Oh, just so you know, they're opening 2 vaccine clinics tomorrow and Saturday in Westchester." So I was telling Diana in the lab about this, and I was like, "It's appalling. We can't figure out how to deal with smallpox that's been chronicled in the Times about how difficult it has been for monkeypox, how difficult it has been for people to get treated appropriately and tested appropriately for monkeypox in New York City. Like they wait weeks, and yet 1 case of polio, and and 2 vaccine clinics immediately open."
Why do you think there is a big difference?
I don't want to go down that road, because I think that it has to do with the population that's affected.
Okay.
And I think that that's horrible.
Yeah, I think if they vaccinate extensively, there won't be additional cases, but it depends on individuals who are reluctant to get vaccinated. So we'll see what happens.
Right. And I think they're just going in with IPV. I don't think they're going in with OPV.
Really? That's
Yeah,
very
which is not the standard.
No.
And it also means that all the guts are going to still be susceptible. So
Oh,
I
yes.
don't really. What?
Oh, this virus is circulating through the U.S. And if you're not vaccinated, it could get you. So you need to be vaccinated until we don't have any
Right,
more
but
circulating virus, which will be a while, right?
I'm not sure we'll ever not have any circulating virus because we just had that discussion too. Because NPOV does revert, it reverts less frequently, but then it picks up recombinants. And those recombinants, some of them have been shown to have um, neurovirulent properties.
Right.
So, how long are those going to circulate? And what's the probability of them either circulating and picking up other determinants or
Yeah,
changing to
exactly.
be more? Because the determinants that we've attenuated are unstable.
Yes, absolutely.
So
Okay.
it just needs to go through a few more million guts, right? And then
Yeah,
it could be, you know.
yeah. I'm sure we'll have more questions, but here is a COVID. The result of a lateral flow assay is valid for 30 minutes. Sometimes a faint red line can appear afterwards due to contamination. Why is this? Does the binding antibody denature? Do you know anything about that, Amy?
The binding antibody does not denature. It has to do with substrate.
The substrate just spontaneously fluoresces or glows, whatever
There's
Okay.
a chemical reaction. Has to do with the substrate. You see that in ELISAs too. If you let ELISAs, like when we do ELISAs in the lab, if they go too long, the reaction with the sulfuric acid and the hydrogen peroxide can turn the solution slightly yellow, and then you'll
Okay.
get a false positive. Has nothing to do with the antibody.
Thanks again for reluctantly answering my question last week about the biggest SARS-CoV-2 surprise. Does he remember the TWIV later in 2020 about antigenic drift in 20 years of Dutch HIV blood sample? I do remember that, yeah. What about it? What are you thinking, Pete?
I don't know.
I don't know. Yeah, but I do remember it. Okay. Frank is in Maryland on business. I want to say hello to Amy. So
Okay.
you guys need to hook up somehow.
Well, he can email you and you can forward it to
All right,
me or
send it to Vincent
something.
at microbe.tv and I'll forward it to Amy.
Yeah, it's
Oh,
good.
I ran out of characters. That was common cold antigenic drift. Yes, that was we mentioned that. I mentioned it last week. It was a surprise to me. But Amy, of course, always likes to remind me that we just never looked, which is absolutely right. Yep.
How we just talked about that too today.
Diana, first time I catch a live stream. Welcome. Glad to have you.
Cool.
I saw you called out everyone's favorite rapid antigen test salesperson. How come you decided to engage on Twitter? I don't know. New York. I have no idea. I was on the train reading. Amy had sent me the article about how endemic COVID looks grim. And I just read his quote and I just didn't like it. It's just misrepresenting what's going on. So I decided to mention it on the tweet thingy. And I don't do that
On the
very often.
tweet thingy? On the
I
tweet
don't,
thingy?
on Twitter. I don't do that very often.
Is that like the ticker?
I'm not going to be
Is
there
that
engaging people. I just wanted to put out my thoughts. I have 136,000 followers. I want them to know what I'm thinking. That, mutation rates in viruses don't change because of selection. Selection is what produces the final results, but the mutation rates are the same. And he's getting it wrong. He's misleading the journalist, and the journalist is misleading people. And I think you ought to understand what's going on. All right. So I thought it was a good teaching moment. But that's it. I put
is
it
a
there,
good
and
teaching
I don't go
moment.
back. I have nothing personal about it with him, but he just should. He knows better. I know he knows better.
Really? I'm not clear that he knows anything.
You think?
Well, many of his quotes are not correct. So every time I think he knows something, he opens up his mouth and releases all doubt that he really doesn't.
So the gist of the article was that, a lot of people are dying, and this is going to go on for a while until we have a complete immunity in the population, which is acquired at a younger age, because if you get infected older, you don't have a long-lasting immunity. But they failed to point out that if, as Daniel Griffin said last week, if you treat people early, as soon as they're positive tested and they're at risk, if you treat them early, you can save their life. And he said it again tonight. We recorded the clinical update. And he also said the vast majority of deaths are still in unvaccinated people. So that article makes you all upset about doom and gloom of people dying for years and fails to point these very basic things out. And I was going to email the author, but of course there's no email. So I just picked on one thing in the article on Twitter. I didn't feel like writing a bunch of tweets about what was wrong with it. All right, so Frank is at a meeting and nobody's masking. So the ASV meeting, we had to mask. So all the sessions we were wearing masks, and I didn't particularly like it, but they asked us to, so I did it. However, on Tuesday night, I went to the banquet, and then afterwards there's the traditional dancing to a DJ who wasn't very good, but okay, why please me? And none of the young'uns, there was a scattering of old people, like me and Kathy and the president, Felicia, and a couple of older people. Nobody wearing a mask on the dance floor. And when you dance, you're pretty close together. So I said to Amy this morning, if I'm going to get COVID anywhere, it's going to be there. Right, Amy?
Yes. But you also sat at an overcrowded dinner table.
Yeah, so the thing is, during meals,
where you learned that you had you were separated at birth from someone in San Francisco who was born in Argentina. I'm not clear how that works.
I was joking. Come on, he was joking.
I know. But now
so
I
anyway,
get to
Frank,
tease him.
Look, Frank, if you're worried, as soon as you test positive, take Paxlovid or monoclonals. But you may not be eligible, right, because you're a young guy. But that's the solution to protecting the vulnerable. Carol enjoyed TWIV with Paul Offit. I asked him if repeated vaccinations cause autoimmunity. He didn't answer the question. I actually asked him twice. And he said, I'm not worried. I'm really worried about original antigenic sin or imprinting. That's what he said. So we're going to talk about autoimmunity on Immune at some point. Are you worried about autoimmunity, Dr. Rosenfeld?
No.
Okay. That was quick.
What's quick?
You just said no. All right, so Frank has a question. Is BA5 more fit or more contagious? Hafiz said more contagious. I am confused. Does COVID linger in the air like measles in most situations? So no, it doesn't linger in the air like measles. It is more fit. And we don't know why. It may be because of immune evasion, and that may drive it through a population. That may make it more contagious, but we don't know. But, there are also behaviors going on now that are unique, right? People are hanging out in the summer. You see all these meetings where people are not masked, and that drives infections as well. So you shouldn't just – we've been arguing for 2 years, Dr. Rosenfeld and I. Don't just say the virus is more contagious. You got to take into account human behavior. But the epidemiologists just take into account human behavior. I don't want to pick on them anyway.
They negate human behavior.
I don't want to pick on them. Billy Bob, hope you liked my song, Clinical Update 94, version 4. Where can I hear this song? Billy Bob, love to hear it.
You didn't send it to you?
He might have, but I didn't see it. Okay, are you guys concerned with developing MS later on after contracting EBV? So this is an interesting question. I spoke with a neurologist from Albany at TWIV.
I told you the exercise.
Well, he did tell me the exercise. But here's the cool thing about TWIV, doing TWIV and live streams and videos online. Everybody comes up to me and talks. And I love it because I learn so much. A friend of Amy's actually said, it's really gracious of you to talk to everyone. Well, I wouldn't not talk to everyone. It's like I Why not? It's what I like to do. And this physician, this neurologist said, am I talking too much?
No, no, no. I
I
just
thought
like
you
to
were my
tease you.
supporter. Why are you making fun of me?
I'm not making fun of you. I'm just teasing you.
All right.
God, get
So
a grip.
he said, I said, what do you think of the EBV MS connection? He said, I knew it 20 years ago. I said, why? He said, I have 1,000 MS patients I'm taking care of. And I ask every one of them, have you had EBV? And they said, oh, yeah. And when did you have it? They all had it later in life. And he said, most of them, it put them in the hospital. So he thinks, severe EBV is what's associated with MS. So, I don't know if that's anecdotal or what, but I'm not worried, no. I think life is too short to worry, frankly. Is that a good philosophy, Amy?
Yeah.
And here's Doreen, of course, who I showed you a picture with us before. Hello, and thanks again for lunch. Polio case, an unvaccinated adult.
Yes.
Is this one of the New York City ethnic groups, vax hesitant? It's in an Orthodox community, right? Isn't it?
I don't know.
Actually, we don't know. We just know that the person, they didn't say that. That's absolutely right. So I
won't be surprised.
Yeah.
I won't be surprised where it is. Excuse me.
What's the advantage of receiving a fourth mRNA dose? All right. So, first of all, so if you're immunosuppressed,
wait a minute. It depends on whether or not you're what kind of immunosuppression. It's not all immunosuppressed people. I sent you the
some immunosuppressed people, a fourth dose sometimes give you a response, right?
Sometimes.
And then elderly
a lot of immunosuppressed people make great responses. You don't see a ton of,
And then if you're old, because you get a vaccine dose, you don't generate much memory. So once those antibodies and T cells go down, you're out of luck, because if you get infected, you're not going to make much of a memory response. So continued boosting takes care of that. So you ask, do you have to do that for the rest of your life? Well, Amy, you'll hear Pamela Bjorkman's, and you sent me her paper with the mosaic nanoparticle. And her approach was really clever, and they get induction of antibodies, neutralizing antibodies against very diverged coronaviruses with that approach. And I think that could be the solution. She's actually putting it in people. They got funding from CEPI, she said.
That's great.
People, it's really good. So that's the advantage. If you're older and at risk, then it overcomes the deficiencies in memory generation. Oh, Vanity is here. Did I say thank you? Yes, I think I did. I gave Vanity some work today.
Yeah, she should be doing the newsletter. It needs to be crossing my plate by next week.
Your plate? Are you going to eat it?
Yes.
Okay, I hope it's tasty.
I'm gonna add it, some salt, some pepper, put it on my baguette.
All right, Kat says, can you explain the functional difference between flu strains and SARS-CoV-2 variants? All right, let's take that part first. That's a good question. So they're very similar. As Ron Fouché said last year on TWIV, was it, Amy? Ron Fouché came on?
2 years ago.
2 years ago, he said they're very similar. They're antigenic variants that displace each other with some periodicity. And in the case of influenza virus, the strains displace each other because one is more fit than the other as a consequence of antigenic drift. They evade antibodies, basically. And for SARS-CoV-2, the only variant that has been really convincingly shown to do that is Omicron, BA.1.2. So the others, I'm not as convinced that we know why they displaced each other. But you see, Omicron is continuing to diverge, and those additional amino acid changes in the spike lead to even reduced neutralization. So there's some similarity, but not completely.
Well, the others diverged because they evaded the immune response. Some of them had alterations in like ORF6 and ORF9, which are immune evaders.
All right, then the
So,
second question is how the vaccine works against variants not present in the vaccine. Well, that's due to somatic hypermutation. When you get a vaccine several times, your antibody genes can make new antibodies that they haven't even seen. That's the beauty of our immune system. Mutations are introduced into the antibody genes, which give rise to altered antibodies, and then the best ones are selected out. And some of those recognize things you've never seen. It's absolutely amazing. You can immunize animals with synthetic chemicals that they've never seen, and they can make an antibody response against it. It's really, really amazing system that's evolved. Do you like the immune system, Amy?
yeah, I do. It's good.
Can you say more about viruses that confer heat tolerance to plants like you were discussing with Rich? Actually, I didn't even discuss it. I just said, some plants get thermotolerance from viruses. So the best example is from Marilyn Rusink's lab. By the way, Amy, I saw Marilyn at ASV. She wrote another book.
Great.
And she's going to send me a copy of it. And so maybe we could have her on and talk about it. What do you think?
I think it'll be great. For sure.
So Marilyn did an experiment in her lab with a grass. It's called panic grass. It grows in hot areas like next to Yellowstone National Forest where the hot springs are. Lots of grasses can grow there, like 55 degrees centigrade. So you bring it in the lab, you can show that these grasses grow at high temperatures. And it turns out the grasses have within them a fungus, and within the fungus is a virus of the fungus. And both of those are needed for thermal tolerance. If you take out the fungus, if you take the virus out of the fungus and leave the fungus, there's no thermal tolerance. And so, very nice description of this tripartite symbiosis. But she believes that there are many other examples. And in fact, she thinks wild plants have viruses of various sorts that confer thermotolerance, nutritional tolerance, and so forth. And, our monoculture crops don't have viruses, so they don't do as well in adverse conditions. So it's really interesting. She worked for many years in Africa trying to get people to grow crops that are more wild than domesticated because of that, right?
Mm-hmm.
On TWIF's 917, Paul Offit said Novavax uses the same adjuvant as Shingrix, Matrix M. People often react strongly to Shingrix. Curious, since the same adjuvant, if Novavax would cause similar reactions. I don't know. Do you know what the adjuvant is in Novavax?
No.
It's something that requires something from a tree that's hard to get, right?
I don't know.
Let me look it up very quickly here. It's made from tree bark. What is the Shingrix adjuvant?
I do not know.
I thought it was something different. It is. The adjuvant in Shingrix is an extract of the Chiaja saponaria tree, according to the FDA label. Wow. So maybe it is the same. So, yes, I would... it could be also the protein that's a major driver together with the adjuvant. So I wouldn't necessarily say for sure, but it could be. Do you know, Amy, if any of the trials with Novavax, they had some...
I haven't heard anything.
Mm-hmm. Very interesting. My sister was sick last week. Nice horse. Same exact symptoms of when she had COVID. Took 3 home tests, all negative. 2 different manufacturers. Could they have been wrong? I don't think 3 from 2 were wrong.
Well, first of all, there's a lot of respiratory viruses that have exactly the same symptoms.
Yeah. Yep. So that's what Daniel's always saying. There are many other viruses out there. So as Amy says, she probably had some other virus. And, many symptoms are similar.
Mm-hmm.
Would an EBV antibody IgG range of over 600 with the standard range of 0 to 18 indicate a recent infection? Well, I wouldn't say recent. Recent would be IgM, right, Amy?
Yes.
600 is pretty high above the standard range, so it could have, yes. So, it's takes months for antibody levels to go down, so it could be recent. Could be within 6 months, yeah. What do you think, Amy?
Could be. I mean, I don't know. I'm not a big proponent of quantitating antibodies like that. I'm trying to make a conclusion.
Polio back in the news. Hope media have Vincent on speed dial. Nope, no Vincent on speed dial. But they should really put Amy on speed dial. But she's with the FDA, so she's kind of conflicted, right?
Why do I need to be on speed dial?
Because you're brilliant.
Okay. They could speed dial all they want me. I don't know what number they're going to speed dial.
You're not
So I
gonna
have this
answer.
phone.
Is that what you're saying?
Well, I have a phone. I don't think it doesn't have a dial tone. It says it's plugged in, but it doesn't have a dial tone.
It's an old saying.
And number two, I know. Another thing is I have no idea what the number is. I don't know.
Your phone
anybody
number?
knows what I don't think anybody knows what the number is. It's kind of like nobody knew the password to my PIF card, even though that there was one.
Okay.
Happens.
All right, here's one for you, Amy. Any idea why some viruses cause encephalitis?
Well, sometimes they target the membranes.
Encephalitis.
Some of it is
what is
swelling of the
encephalitis,
brain.
Amy? Can you define it?
Swelling of the brain.
It's inflammation,
swelling of the
right?
brain.
Inflammation of the brain. And
And
the brain,
sometimes,
the cortex, not the meninges, because that would be meningitis, right?
right. Sometimes it's infiltration of a lot of water due to immune responses and stuff. There's a lot of reasons why you get encephalitis.
But you have to have initially some virus there to trigger the immune responses, right?
Yeah, for sure.
Oh, look at this. Peter is in Buyukada Island in the Sea of Marmara near Istanbul. Wow, that sounds exotic and hot. Hope you're enjoying yourself. Thanks for listening from an exotic location.
Yeah.
Barry says, could a virus replicate so slowly that previous vaccinations would prevent transmission since T cells would kill infected cells before enough viruses would be produced? Yeah. I suspect that happens with many of the viruses that are in us all the time. Yeah. They're at very low levels. Yep. It's a good thinking, Barry. If SARS-CoV-2 still spreads in vaccinated individuals, thus creating mutations, how does vaccination end the pandemic?
Because the pandemic's only concern is the development of severe disease that overwhelms the hospitals.
Yeah.
It has nothing to do with anything else.
And so with increasing immunity, there's less and less severe disease. Now, the unfortunate thing is that older people are going to have to be vigilant, right, because they don't develop memory very well. So
they develop memory.
Not very well, not as good as a young person like you, Dr. Rosenfeld.
They were going to die anyway.
26-something-year-old like you.
I'm not 26 quite yet.
Oh, when are you going to be 26? Next year?
I don't know. I'll let you know. I'll let you know.
So, but first of all, polio vaccination blunts transmission for sure. But as Amy said, the main result is it reduces severe disease. And really, that's what your concern is. Because as Amy will say, there are many, many, many rhinovirus infections. But it's not too severe, right, Amy?
Right.
So we don't consider it a pandemic.
It depends on what serotype you're talking about,
right,
or genotype. Are you talking about C or A or B? Because B, we don't really think that there's any disease to.
Right, A and C mainly, yeah.
A and C, and there's some cross-reactivity with A and C, so depends.
And you can look at the common cold coronas, right, which infect most people on Earth, and very little disease. People,
some people do go to the hospital first.
Yeah, So Paul Offit said it accounts for 15. The common cold coronas account for 15% of childhood hospital admissions. He said for like bronchiolitis. So that's a substantial amount. But
Right, but it's not. But when you put it in perspective with how many kids go to the hospital with RSV, it's got to be
many,
like twice. It's got to be twice that
yes,
at least,
yes,
right?
anymore, yeah.
And the how many adults go to the hospital with common cold coronas? Uh, and this was another point about RSV. Remember, we had this conversation when I said that older people are huge population drivers of RSV,
Mm-hmm.
and you said no. But how many older population come in with RSV?
It's substantial, yeah. And it's because I wonder if they saw it in infancy or childhood at all. That'd be good thing to look at, because if you got it later in life, then you're not going to make as good a memory
Well,
response, and that could be an issue.
I bet you that the immune response is not lifelong.
It could very well be, yeah. Sorry to sound like a broken record. Thai study in JID concludes breakthrough infection produced a comparable immune response to a booster vax. Did Amy say you think?
Yeah.
She's always said it's comparable, yes. And I always tried to kind of say maybe not, and she was very adamant in her position. One thing about Amy is very secure in her scientific positions.
Sometimes.
All right, so this is Amy is AMY, but you could change your name to AIMEE, wouldn't?
I
That
had
would
a
be.
friend who, so I had a friend in high school whose name was Amy, and she was born the day before me.
And this was spelled like this. Anyway, DGH says, I'm wondering what a normal workday is like for you. You seem to put in way too many hours.
Okay. Can we not discuss this?
Yeah, it's fine. I just wanted you to show that people are concerned about you.
I understand. I greatly appreciate it.
But I have to say that when Amy was in the lab, I never told her not to work. Right? So
You never told me to work or not to work. You didn't say very much about my work ethic
no, I
or
left you
my.
alone. I left you.
Yeah,
That's my.
exactly.
That's the way I work. My managing approach is to leave people alone. Make sure they have the tools they need and leave them alone. Right? Wouldn't you say that's true?
Yeah.
And some
So,
people don't work under those conditions. They need a lot of attention, and so they don't do well in my lab. It doesn't matter
yeah.
anymore.
Whereas if you were constantly attention, it would be overbearing and I wouldn't work well whatsoever. I'd be like, stop nagging.
Well, that's what you'd say. You'd say, Vincent, stop nagging me. I can handle this on my own. I say, okay.
Yeah, exactly. Why are you micromanaging?
All right. Hans wants to know,
Isn't that
Amy,
my
and
next favorite one?
this is for you. Immunodeficient people are at risk of ICU death and should receive a fourth dose. But what exactly are these immunodeficiency? All, for example, obesity is an immunodeficient state, and 40% of people have it. So you said before, some immunodeficiency people make a good
Yeah,
immune response.
B cell people don't seem to have a deficiency.
So
Like, yeah,
people with B
many of
cell
the
deficiencies seem to do well with COVID, right?
yeah,
However,
they do
people with
well with
T cell deficiencies don't have such a good
right,
time, right?
right.
And obesity is a problem, right? That's a risk factor.
Yes, yeah. But obesity is a hyperinflammatory response too.
Right. Has
And
anyone been?
people who have interferonopathies are not good.
Oh yes, that's bad. Yeah. Has anyone been playing Redactyl? So I really liked it when I went over it during the TWIV, but I haven't touched it since. I just get
What is
distracted.
redactyl?
It's a game where they give you a paragraph where most of it's been redacted, right, blacked out, and they give you some clues, and you have to guess the whole sentence. I think you'd actually be very good at it, but you're not a gamer. No.
No,
I don't
not a
see.
gamer.
I don't see Animal House tonight. Is that her name?
Animal Party. Animal Party.
Sorry.
I don't know, but I see Richard is here. But I don't see Animal Party. Maybe she's working. Maybe she stopped listening. She wasn't here last week either. I don't think. Or maybe she was here last week, but not the week before. Plus, she's got bees. She's got to take care of the bees and the boys. She's got the BBs.
So Pete says Fox has this idiot doctor, Mark Siegel, on stating that they should have given Biden bebtelovimab and Paxlovid. He needs to read the NIH guidelines. Yes.
I don't think it's the NIH guidelines. I think it's the CDC guidelines.
Isn't it FDA? No?
CDC guidelines for treatment is what Daniel always quotes.
But there's an example of someone who's fully vaccinated, but he's older, so you give him Paxlovid at the first positive. That's the way to do it, and he'll be fine.
Yeah, but you didn't. But that doesn't mean he would have automatically developed severe disease. He has a higher probability of it than you or me do. But it doesn't mean that's what he would have gotten.
Of course, but you don't wait to find out. You treat immediately, because if you wait, then it's too late. That's the whole point. Many doctors say,
I know.
You seem to be okay. Let's wait and see what happens. And Daniel says, no. And then it's too late, and Paxlovid will not help you. And that's why people are dying.
Nothing will help you.
Well, Amy could, but you know.
I work on different things.
Frank's in Rockville.
I know where that is. He's only here tomorrow. Till tomorrow. It's right up the street from me. I know where that is. It's up Wisconsin.
Email tonight, Frank, and I'll pass it on to the good doctor. Okay? Vincent at microbe.tv. Let's see if it's there already.
Maybe
not there yet. Okay, here's another one. Do you think we should have mass vaccination for monkeypox? I think we should have ring vaccination. I think we should have ring vaccination. We should vaccinate everyone who's in contact with a case.
Yeah, for sure. But we're not there for mass vaccination quite yet. We might get there, but I don't think so.
Would you say the monkeypox vaccine is as safe as the COVID vaccines?
So this guy has an interesting handle. Look, it's Polio Pete.
That's good.
I wonder if he had polio.
I don't know. Polio Pete. Why is your handle Polio Pete? Tell us. If you're willing. You don't have to, of course. First of all, it's a smallpox vaccine. There are two of them. And one of them, ACAM 2000, is vaccinia virus, which has more side effects. And I would say it probably has more side effects than COVID vaccines, except for the more serious ones, right? Like the myocarditis and the clotting. Those are very serious. And then the other smallpox vaccine is a vectored vaccine, which is not vaccinia, so very few side effects. So that would probably be as safe as the COVID mRNA vaccines.
I think they're safer than the COVID mRNA vaccines.
You think?
I think so.
Okay. So here we go. We're going through the award. Okay, thank you everyone for your.
See, everyone's proud of you. They're all happy. It's good.
Thank you. Well, this is our crowd here. We've been hanging out with these people for the last 2 years, and I think there are very few new people here. Although I saw there's some.
I don't know. I don't know who this Nancy Gregg person is. I've not seen her before, but I'm glad to have her come.
How serious is monkeypox for the general public? Does it transmit through the air or on surfaces? No, not through the air, not on surface. You need to have intimate contact. Okay, you got to have skin touching skin. You don't need sex, but you can have intimate contact. And how serious? There are very few deaths from this monkeypox outbreak. But we don't want people to get it. Why take the risk, right?
Exactly.
Steph said there are bodies attached to the TWIV heads. Yeah, we all have bodies. That's right.
Oh, really? Not a bobblehead doll?
Is Moderna developing an mRNA EBV vaccine? I don't know, but I bet someone is. Maybe not Moderna.
Yeah, they are.
Are they?
Mm-hmm.
Okay, Amy knows.
It's on their website.
Okay. Amy, do you have an eidetic memory? What is an eidetic memory? Do you know?
Like
Okay, Frank just sent the email, so I'm going to forward it to
So
you.
yeah,
Should I
forward
do
it
it
to
to
my
your Gmail?
Gmail. Yeah, forward it to my Gmail, and then I'll contact him. I don't know. I don't think so. I don't think I have a eidetic memory, but I don't know.
I don't have your Gmail here. I don't want to give it out. I'll get it. Don't
Do you
worry.
want me? That's fine. I'll
I'll
text it
send
to
it
you.
to you later. Do you have an eidetic memory? So what is that?
I don't think so, but I don't know.
Relating to or denoting mental images having unusual vividness and detail. I don't know. I have no idea. I can't answer for you.
I
Right?
don't know how to answer that question. How would I know? It's not like I tested it for it. I tested my memory for it. I don't know. I'm just me. Why do I need to have a label? Can't I just be me? Are you listening?
Yeah, I'm listening. You know, I always have your devout attention. You always have my devoted attention. Okay, Amy dot. Okay, I got it now. Amy at Gmail. Okay, I'm forwarding you this. Let me know if you get it.
Okay. I'll
Good.
let you know.
You guys can hook up. All right, back to the questions. What's the virus on the standard TWIV mug? I think it's the same as this one. It's polio virus with the receptor bound. You know, so it's kind of an artificial situation because these are all in blue. These are soluble receptors. It's like the model that I have behind me. I'm not going to get because last time I did it, Amy was unhappy. You said put the model down. Do you remember?
Yeah, probably. Yeah, probably. That sounds like me.
Oh, here's one for Amy. How does identifying viruses from metagenomics data work? I got the herpes virus keychain. That's not an answer. Tell them how it works.
So they sequence and then they make overlap. They sequence so that they have overlapping pieces, and then they try to piece it together based on overlapping sequences, and then extend. And then they go to the next end and find a piece that overlaps it, and then they build upon it to get the entire genome. But whether or not that's infectious virus and whether or not that actually is virus is a little iffy.
Yeah,
Because it doesn't
usually have
tell
short.
you that it doesn't tell you that it actually makes anything.
So, do you typically have short reads?
Yeah, you typically have short reads. So you do like 150 where 30 of each end you aim for like 30 of each end to overlap with another piece so that you can build it together by the overlapping pieces.
Into what's called a contig, right? A contiguous
Yeah,
sequence.
a contiguous sequence, yes.
All right. Farhead said, "I'm not convinced why we shouldn't have a cocktail of variants, because eventually you keep getting changes. You got to look at Bjorkman's approach. It's way better. She figured out a way to get to immunize so that you get antibodies against the more constant epitopes. So there's 4 antigenic sites, 4 major antigenic sites on spike, and 3 and 4 are very conserved. And she figured out a way to get antibodies because they're immunosubdominant. So she figured out a way to do that. I think that's the way to get, because it covers you against future variants. So the next TWIV will have that, which will drop on Sunday. That's the TWIV from ASV.
Mm-hmm.
Congrats on the excellent TWIV where a study on mosquitoes and acetophenone was done. Why can't they do COVID studies this thorough? Because they're rushing it. Wouldn't you say, Amy, they're rushing it?
Yeah, they are. Why would you do it? Yeah.
I mean, that study in mosquitoes took well over a year, I'll bet, if not many years, multiple years.
And many years.
People don't have the time or don't want to put in the time for COVID. They want results because they think they're going to save the world.
Mm-hmm.
Have there been?
Well, you needed to do something for the pandemic at the beginning, right?
Yes, and That's been done, isn't it? Then we have vaccines that work, right? But I would argue that improving the vaccines is a good thing.
Yeah, but I don't think you're going to improve them by using the same platform. You have to change platforms.
Well, I think that the nanoparticle that Bjorkman is proposing is really cool. She says it makes a mosaic of RBDs, receptor binding domains, of 8 different
but
spikes.
it's still based off of only spike, and you're only analyzing IgG and stuff. There's a lot of other stuff. So if you make a live attenuated vaccine, you're probably going to make it the best.
Could
And
be.
infectious attenuated is probably the best.
But you're still going to get antibodies to the dominant epitopes. That's the thing about this. It really biases it to the subdominant epitopes. Anyway.
Well, the subdominant epitopes, because that's the only thing. But it's just like, whatchamacallit, like making antibodies against the stock portion of flu.
Right. Okay, have there been studies related to long COVID in infants? We've kept our 1.5 protected because we're afraid of the long-term damage. Are you aware of any long COVID in infants? No? Okay. What was I going to say here? I wanted to see. I don't think Daniel has talked about it. Let's see. When I am in doubt, I check for. Actually, a recent study out of Denmark. Yeah. Is they look at long COVID in infants. So this paper in Lancet Child and Adolescent Health, 44. What?
I was wrong. I didn't know that they had done it.
44,000 children in Denmark ranging in ages from 0 through 14. 11,000 tested positive. At least
tested positive.
And the children who tested positive were more likely to experience at least one symptom for 2 months or more than those who never tested.
What's one symptom?
Most common symptoms varied by age. For children up to age 3, it was mood swings, rashes, and stomach aches. 4 to 11 experienced memory and concentration. 12 to 14, it was memory and concentration, mood swings, and fatigue. Children under 3 had seemed to have the most problems compared with those children not diagnosed. So yeah, it's so the thing is overall, the incidence is lower, but they can also get long COVID. But how long
I just
it lasts,
don't
we
understand. Well, the other thing is I don't really understand what is how you're measuring long COVID. So a rash, lots of kids get rashes. You generally get like a prickly heat rash for a really long time when you're an infant. And 3-year-olds are not really considered infants.
Yeah, I agree.
3-year-old is not an infant. And then if they have stomach aches, a lot of children have diarrhea when they switch foods and go on to, if they were breastfed, go to, what's it called? Formula, and some formulas don't always work out well. So I don't really know how because it seems to be like when you talk about long COVID, you're reporting fatigue and brain fog and other stuff. So I don't know. I don't really understand.
They said any symptoms lasting more than 2 months after symptom onset, and which is that's also, as Paul Offit said, a difficult definition. So I think we're still waiting for a good definition of that. Have most people been vaccinated for polio in the U.S.? Yeah, right, Amy? The vaccination rate is high overall, but there are pockets of lower vaccination rates, and that's where you can get these cases like the one we have. Do you know what town in Rockland? I don't know. I haven't released
It's not Rockland. It's Westchester.
Westchester. No, it's
So
Rockland.
Rockland is a county. No, it's in Westchester, I think.
Well, let's take a look.
Oh, maybe
Polio.
is it? I thought it was Westchester.
I thought it was
Maybe it could be
Rockland
wrong.
County. Yeah, it's Rockland
Okay,
County.
Rockland. Okay.
But they haven't said what town. No. What happened to the TB guest? I was on TWIM. That was Sharon Darwin, and she was on This Week in Microbiology. And it was
Yeah,
like
at ASM.
AMSM just a couple episodes ago.
Mm-hmm.
Westfield, thank you for your contribution. Erosion of trust in institutions continue in LA. Public Health Director threatening mask mandates while USC hospital MDs saying no emergency in their hospitals. Anyone following science anymore? Any comments, Amy?
The right hand not talking to the left hand, and the left hand not talking to the right hand. What do you want me to say? I don't know what the situation is in LA. I wasn't really following it that closely, so I don't know.
If even Paul Offit said he does wear a mask in the hospital, which seems to me to make sense, right? Because there are sick people there and many immunocompromised people, so it's weird. Is OPV cheaper than I would say? What is cheaper, OPV or IPV?
OPV by bucket loads. It's
Buckets.
like 10 cents. It's like 10 cents versus like a $2.99 or something.
So that's part of the reason it's used in many countries, but also it's easy to give, right? You just drink it. And it's really good at stopping outbreaks. So, and IPV has to be injected and it's more costly.
Yeah.
Very concerning that people don't get vaccinated. We had measles in Michigan from a person visiting in New York, tracked, traced. We got info on every store he visited, etc. It's very disturbing, especially for polio, where we know the vaccine will prevent disease. There are no negatives to having IPV. I don't see why you wouldn't get vaccinated, or measles vaccine. Did polio took how long to hit the news? A month. So the case was a month ago, but as Amy said, they misdiagnosed it, and they only did the PCR in the last few days, I think.
They didn't do PCR. They sequenced. They sequenced the whole genome. They didn't do
Well, you have to amplify by PCR, don't you? To do deep sequencing,
Not
right?
necessarily.
No?
Can, depends on what technique you use. You
Okay.
can chop it up. But you have to RT it. But really, they did next generation sequencing.
And I think the sequencing was recent, right?
Yeah, the sequencing was completed just this week.
That was
maybe
done up at the Wadsworth Institute. Yep.
Yeah, in collaboration with the CDC.
I think they both did it independently, according to Daniel. Anyway.
I'm not sure. When the woman discusses it on Twitter, you can ask her, because I believe I was told by somebody that it was only the CDC, but I know that Wadsworth was involved.
I want to have a neurologist who was involved talk on Twitter, but she said she can't talk right now. Next week. It's okay. It's fine. I don't want to get her in trouble. Did you get my questions? I will look at it again and get back to you. I'm sorry if I didn't answer. I get a lot of them, and sometimes they get lost during the day, as Amy will attest.
Yeah.
Thank you, Morris, for your contribution. Really appreciate it. And Steph says, Amy, same issues occurring in San Francisco with affected population and monkeypox.
Not surprising. Actually, it is a bit surprising because they're usually more liberal.
Philip had polio vaccine about 1960, OPV 92, IPV 2022. It should be good.
Yeah, for sure.
I had OPV. I had
Why
IPV.
Why did he have IPV and OPV?
I don't know.
So late.
I could guess, but Philip, tell us why you got. That was so recently. Maybe travel? Could it be travel, Amy?
Could be. I guess. I mean, I don't know any place that requires a polio vaccine for travel.
Yeah.
But it could be. I don't know.
Laura says, I'm hearing of people 60 up getting mild symptoms for 2 to 3 days. Those feeling terrible seem to be younger. Any data suggesting elders tend to get BA5 less harshly? No. I haven't seen anything convincing. I think it's just the same as every other variant. I don't think there's any difference. At least the data don't say that. Have you seen any, Amy?
No.
And Margot says, that's exactly what I was going to say, Amy, about why the delay in monkeypox. Right. We talked about that. Is there any point in spreading out a pediatric COVID vaccine with routine immunizations besides tracking side effects?
Wait, I don't understand the question. So they don't think you should...
Kids get routine
so they don't think that they should incorporate the vaccine as a regular childhood disease, as like a regular childhood vaccination?
Oh, do it with the routine immunizations that kids already get.
Yeah, that's what I'm saying.
No, she said, why shouldn't we do that? Is there any reason not to do
should.
it? I think it's fine,
We
yeah.
should.
I
It's
agree.
more than fine. It's the smartest thing to do.
Is it possible that the presumption that monkeypox is limited to a given population is misleading since others may not be a focus of testing? Yes, Daniel said that tonight. He said he recently tested a woman and she was positive. So he said we have to expand our testing. And so testing is getting much easier to do now. And so hopefully that'll do it, because it's hard to believe it hasn't spilled over into other populations, right? What do you think, Amy?
Yeah, I always said it was hard to believe.
Yeah. Was the polio vaccine we had as children in the 60s or late 50s still protecting us? It's protecting me. Of course, I worked on polio, so I probably immunized myself.
I was going to say you have like a
every day.
gazillion boots.
Do you want some of my serum, Amy? You want some of my B cells?
Sure. Don't you think mine are pretty B-celled up too? I work on a lot of polio.
Well, I mean, I know people who have gotten many doses of polio vaccine. Maybe they would be better than me or you. Yeah, your B cells are pretty good. Them. Your B cells.
I was going to say, I can always get that person to donate, so I don't need to worry.
I think it's
and his brother could donate too.
I didn't know he had a brother. I think, Markle, that yes, the polio vaccine is long-lived in its protection. Yes, it's very good.
He said it was for travel. That's interesting.
Ah, cool. Do you think vaccines are prioritized when the disease affects children more than adults or special populations, i.e. polio versus monkeypox?
Yes.
Really?
Mm-hmm.
So we're quick to take care of kids?
Yeah.
But that wasn't the case with COVID, right? Took care of adults first.
It was the population that was, it's odd that we cared so much about old people dying.
Well,
it was only because it was in such a mass that it overburdened the hospitals. Otherwise, I don't think we would have cared.
Remember, there was the talk for a while that we only make vaccines first in adults, but obviously for pediatric vaccines, that's not true, right? Do you remember that narrative?
Yeah, I do, but that's not true at all. Didn't the chickenpox vaccine come before the shingles vaccine?
Yes, it did.
Okay, right there. That's not true. But yeah, if SARS-CoV-2 infections hadn't generally hit the older generation and overburdened our hospitals, we wouldn't have made such a big deal about vaccinating the elderly.
You know, if you look at the history of vaccines, most of them are childhood vaccines, right? I mean, there are adults. We got influenza vaccines. We have hepatitis vaccines, yellow fever, but many of them are childhood as well. Yeah.
Most of them are childhood.
Professor Cleona O'Farrelly in Trinity College is studying people who shared a bed for several nights with someone who had tested positive for COVID, but who themselves didn't become ill. Yeah, I'm not surprised. I think some people just don't get infected for whatever reason. Maybe they have a great innate immune response. I've worked as a public health physician in the Ministerios de Salud de Costa Rica for 25 years. We use IPV, no longer OPV, just for the record. Great. That's good to know. I think it's good to use IPV, and the more countries that do, the less we're going to have circulating vaccine-derived strains, right?
Well, who knows what you're going to have? Because your guts are fully susceptible.
Yeah, Jeff, we've been talking about polio case on and off. Yep. Do you think there could be a correlation between people who have more symptoms after the vaccine and people who have worse cases of COVID? Maybe some specific characteristics.
I don't know. Some of the people I know who got ill off of COVID did not appear as sick from the vaccine as I did.
Yeah, I don't think there's Daniel has said there's no correlation between vaccine reactions and severity of COVID. So I think not. I know it's a toxic environment on Twitter. We need the right information, or at least more correct. I understand. I prefer to provide that information here with my colleague, Dr. Amy B. Rosenfeld. And
Amy B. Rosenfeld. Oh, my God. My whole formal name. My God, what have I done?
B standing for Beth, right?
Yes.
Is that like a biblical name?
No. I don't know how she got to Beth. Amy is for amor. It almost in every language it means loved or beloved or has some connotation, but that's what it's from.
Anyway, here I lost my train. Here or on Twiv and the other pods, I prefer to put my good information there in my lectures and so forth. I don't want to get into arguments with people. In the past, I have tried to correct things, and people tell me I don't know what I'm talking about. I'm wrong, and that really pisses me off because either I or Amy will let you know when I'm wrong. Okay, not you. Amy has
most
earned the
of the
right.
time, the people.
Amy, you have earned the right to tell me I'm wrong.
Okay, fine. But most of the people who tell you that you're wrong on Twitter don't know anything anyway. They're just big bullies.
I understand, but it still bothers me, and I'm just.
I know. I don't want to discuss things with big bullies.
Yeah. Would you personally get a second Omicron variant booster? Or do you conclude that one Omicron booster gives adequate protection? Well, first of all, right now there are no Omicron boosters, but one is coming in the fall in the U.S. So I don't know how you could get two. But you're supposed to get the two ancestral. If you're new to being vaccinated, the FDA recommendation is you're going to get two ancestral doses, and then you're going to get a booster made up of some Omicron. Right now, they're talking about 4 and 5, right? But who knows what it'll be like in the fall? But I think you start with one. I don't know why you'd want to plan two already. But I think that there's variation will continue, and those will be out of date as well. So, let's see. Vincent, you're a great dancer. You really got the groove. Oh, thank you, Pablo. I'm okay. I just like to get up there and move around, and I like to watch people dancing. You know, Amy, it was funny. I would dance with Steph or Kathy or some other, and then they would
wait a minute.
Kathy Spindler, yeah.
I know. It's just I'm surprised that
she likes to dance. Well, I started by asking.
I'm just surprised that she allowed that. That's all.
No, Kathy really likes to dance. I went to her table, and she was sitting next to Bert Semler. I said, Kathy, you want to dance? And Bert said, good job, Vincent. She likes to dance. So
why
we
didn't
went dancing.
Bert get up and dance with her?
Bert doesn't dance. Bert doesn't dance. Anyway, then she left.
He doesn't.
At some point, she left, and she said, it's not you. I don't like this music. I don't remember. So I was there for a while by myself, and then people will come up and, hey, come dance with my group or come dance with my group.
That's cool.
It's very cool.
Great.
I had a great time. Thank you, Pablo. I'm okay, but I just like music, and I really think other people are better dancers, but I like watching them. Yeah.
The funny thing is that you very rarely listen to music.
Yeah, that's true. And I rarely dance. I dance really once a year at ASV. And
That is true.
Last time Amy was there, she was dancing with Steph, according to Steph Langle from Immune. All right. Why are you not worried about autoimmunity, Amy? Please explain.
Not worried about autoimmunity?
When you get lots of doses, lots of boosters.
So it's not clear to me how much antibody and stuff is really still there when you get all these boosters and exactly what you're doing. And there have been other cases that, while they're anecdotal, that I know about where people have gotten a huge number of the same vaccines and they're perfectly fine.
I mean, it's a theoretical possibility, right? That
right.
Triggering so much antibody diversification could make you make antibodies against yourself.
Right. But the probability of you doing that is very low.
I think so also.
And you select out a lot of stuff that reacts with yourself. And as I said, anecdotally, we know of people who've gotten many, many doses of the same vaccine, and they appear to be fine.
Yes. All right. Now, what's the concern with imprinting? This is not a daft question. It's a good question. So imprinting is when you get the first dose of a vaccine, and then let's say you get a different serotype later on. You're biased in making antibodies against the first version that you saw. And that happens with influenza viruses. And Paul Offa gave us an example where it happens with human papillomaviruses. So,
also happens with dengue.
With dengue. So the original
it's a euphemism or synonym for antigenic.
Yeah, original antigenic sin.
Yeah, exactly.
Yeah, but imprinting is a little more neutral. It's probably better. So that's the concern with
Whatever.
COVID. You have the ancestral vaccine, and then variants arise. You give a variant vaccine, but maybe you're only going to make a response to the ancestral. But it doesn't happen because they've done trials with the variant Omicron-based vaccines.
But it's not even what you see in patients who get natural infections. So I don't know why we would even start talking about it. He would even bring it up with as a concern because it's not what you see with people who get repeated infection with COVID, with SARS-CoV-2.
Yeah, right. But it does happen with other viruses, right?
Right, but I'm not arguing it doesn't happen with other viruses. I'm arguing that you can, by extrapolation, because vaccine is a targeted, controlled exposure to the antigen, which is spike.
Right.
So in a non-controlled environment where you can get reinfections, you don't see the second infection becoming more and more severe. Should have given you a hint.
Yeah. So, okay. I agree. Now, going back to autoantibodies, someone wants to know, what is an autoantibody? It's an antibody against yourself. As opposed to a virus. Sometimes we make antibodies against ourselves, and that's not good because then you can get sick.
It's opposed to anything. Virus, protein, bacteria, trypanosomes, chemicals.
So Gwen's going to a wedding, 86-year-old mom, thinking we both should be masking. If you want, your mother's in the at-risk population where, even with all the vaccine doses, she may get severely ill. So you could deal with that with masking, yes. And make sure you're ready to get Paxlovid should she test positive, right? You go home, she tests positive, get Paxlovid right away. A 94-year-old got sniffles negative test, so I looked up UK Paxlovid. A 94-year-old with diabetes is in high risk. So here, the age alone puts you at high risk for Paxlovid. So that's really unfortunate in the UK.
Not necessarily.
Yes,
So
according to
when
Daniel, yes.
No, he's wrong because when my mother and her husband got infected over Christmas, he's 93 and she has lymphoma. She was considered eligible for Paxlovid and he was not. So it's not what Daniel said is not correct.
I think Daniel is correct. Daniel is a infectious disease physician
Really?
treating
So
patients.
how do you explain the fact that when my mother and her husband got infected, that she was the only one who was eligible, and he's 93 or 94, and she's 20 years younger, and her condition of lymphoma made her eligible, where he was not eligible.
Okay. So
I don't
basically,
think he's 100% correct.
the FDA is it's for patients that are at high risk for progression to severe COVID. Okay, so positive testing and who are at high risk for progression, and then to know that.
Well, high risk, right? But as is wishy-washy, it can be interpreted any way you want.
I'm under the impression that Daniel is always saying, here, all the adults are at highest risk of getting sick, people over the age of 65. So I think that would be covered by the EUA.
It's not.
So why did Anthony Fauci get Paxlovid?
He's Anthony Fauci. Why did Biden get Paxlovid? He's Biden.
Okay.
Let's get real here.
I think that
you know exactly that it's not equal.
But the people who are dying, even though they're vaccinated, are over 75. They should be getting Paxlovid as soon as they test positive.
But they're not always.
All right, but you agree that they should, though, right?
I didn't say that they shouldn't. I'm just saying that it is not necessarily considered a risk.
Okay. Rima wants to know when I'm coming to Iowa. I'll let you know when I'm nearby, right? So I can make a trip to see you because I screwed it up when I went to Nebraska. What exactly defines when a virus branches off to a variant or subvariant? Well, so there are variants and then there are variants of concern, right? So that's what the WHO has given the name to, where they say, oh, these sequences suggest that it might be more severe or antibody evasive, blah, blah, blah. But a variant is anything that's different from another virus. So if I could have SARS-CoV-2 and Amy does, our viruses would be two variants because they wouldn't be the same sequence. Because she would have gotten hers in Maryland and I would have gotten mine in New York. So a variant is just a sequence difference, but a subvariant is a sequence different. Hope you enjoyed Madison. I couldn't believe you're actually in my town. Well, I never left the convention center. Oh, I went to the Great Dane. That was very nice. I walked 2 blocks. But you know, the band played in ASV, Amy. Herpetic Legion.
Oh, really?
Yeah, but I didn't go to see it. It was in a local bar, and it was too late.
Ian Moore?
No, they had, John Parker from Cornell and a couple other people from Cornell. Then they picked up some singers so I didn't go. And no, Ian Moore wasn't there. He was the drummer last time, right?
Yeah, I think so.
Last week, where Amy lambasted Vincent for getting his John Campbell's mixed up, I would definitely categorize this as educational and entertainment. I'm sorry. It actually meant it was John Campbell versus Bill Campbell, right? The parasitologist. Yeah, I mixed them up. I'm sorry.
Doesn't bother me.
Thank you, Westfield, for your contribution. Often said pandemic ends when we say it ends. I went to the amusement park 7 days ago. Thousands, no masking, no distancing. I'm fully vaxxed, wasn't worried. Seems public outside of media, Twitter, and politics have ended it. Yeah, I agree. Because they want to. So, Amy, Steve says, Amy's mom's husband's doctor's internet is broken.
Could be.
They remember what you say. Don't you love it?
I do. It could be. Could be.
What about Evusheld? Yes, Evusheld is a prophylactic, right, that you would get ahead of getting infected, and it
I
lasts for
think
so
they're
many
also
months.
using it for. I think
Yeah,
they're also using it
for
for
treatment
therapy.
now. Yeah, they did a trial and they found that it was worth it for treatment as well. So yeah, that's absolutely indicated for immunocompromised. Yeah, for sure. Remember your sugar cube? How old is old when it comes to needing a fourth dose? I would say 75 and up. Hey, I'm going to need a fourth dose in 6 years, Amy.
I'll worry about it in 6 years, Vincent. I'm not going to worry about it now.
Actually, it's in 6 years we will need a fourth dose.
Exactly. So I'm not worried about it today.
How does asplenia affect the immune system? So you take out your spleen, right? Is that anatomic asplenia?
I think so.
Any impact on vaccine response or risk for severe COVID? I think that's a big deal, right? Not to have a spleen.
No, I know somebody who doesn't have a spleen and they're fine.
What do you mean they're fine?
They got vaccinated and they mounted an immune response and they're fine. They're fully protected.
People who had their spleens removed were not part of the COVID vaccine trials. Here's a page from Newfoundland, information for people without spleens. We do not know exactly how much protection a vaccine will give people who have asplenia compared with people who don't. But it might work I'm sure by now we know what happens when you vaccinate asplenics. Right?
I'm sure. Yeah.
A number of papers showing that the spleen is of prime importance in the antibody response. Anyway, so I don't really know. This is a good question. I'm not aware.
I just have anecdotal evidence. I know somebody who doesn't have a spleen and they've been fine.
Yeah. And I think here's a page with a list of vaccines used in asplenic people, but I don't know. That's a good question. Autobotodogo. Very good. Thank you, Vincent, to assist Amy at expanding her opinions. Maybe many of us would like you to expand on your answers. If she doesn't, I will. We're a good team in that sense. Don't you think, Amy?
Yeah.
When Amy wants to be, what's the opposite of loquacious again?
Verbose.
No. Really?
Verbose is wordy and loquacious is very sparse.
Loquacious is talking a lot.
Oh, so it's laconic.
Laconic, thank you. That's the word I wanted, laconic. I do like those words, but I never remember them. Amy can be laconic, and Vincent is rarely laconic. And Amy does this when I'm loquacious.
I'm just teasing.
But fortunately, I
have a sense of humor.
I have a sense of humor, yeah. Professor Robert Clancy talks about the mucosal chamber, T cells, and gut bacteria. Fascinating stuff for lay people like me. Maybe it's well known by microbiology, virology, immunology students. Yeah, it's very interesting. It's a chamber. I haven't seen it referred to as a chamber, but that's a good way to put it.
What?
The mucosal chamber. You have the mucosal cells, and then you have bacteria there, and then there's the Peyer's patches with the lymphoid cells, and they're all kind of working together.
So
right, the bacteria are stimulating immune
So
responses.
don't you think that this is on any mucosal surface?
Yeah, no, for sure. I'm not saying it's special. No.
I was gonna say, so like your vagina, your nose, your mouth, your anus,
your colon. Yeah, absolutely. So Claire says in the UK you get a fourth dose if you're over 50. Okay.
I think that the United States has changed it to 50 too.
I think that's correct. But I'm not getting it till I'm 75.
What happened to I wasn't getting it for 6 years? You'll be 76 then.
Oh, I'm going to be 75 in 6 years. All right, you think
5 years because you're going to be 70 next year.
I'm going to be 26 next year.
Oh, sorry. So then you got a long way to go to 75 there, babe. You got 49 years.
Vincent, I'm glad you did the tweet thingy because I read that article and it was depressing. I shouldn't be depressed because, as Daniel said, and I really like this, we can save pretty much everyone by rapid response with antibodies and antivirals.
So wait a minute. So how
Uh-oh.
come a few months ago, when I quoted that basically from the Cardiff of the FDA, I got ripped to shreds.
Wait a
When I
minute.
said, he said on Slavic like months ago that there's no reason for anybody to still be dying of SARS-CoV-2 infection since we had antiviral therapies and vaccines. And people like, I said that on this live stream, and people like flipped out and started attacking me. Go back to the archives and you can find it.
Okay. We just took a while to come to agree, okay? You should be happy that
Well,
we're coming around, okay?
I know it's a rapid pace at this 4-month speed. It's kind of like the 37 days it took them to fix my P card, my PIF card. I said to the security person who sees me every day, he goes, "Is it fixed? Is it fixed? Is it fixed?" I said, "Today's the 37th day. It's finally fixed."
Amy is a vibe, and I love that. Sometimes as scientists we have to say, I don't know, and she feels very confident about it. Can you be a vibe or have a vibe? I don't know. Isn't the saying you have a vibe?
I guess.
Happy 26th birthday to both of you. Yeah, we're both 26 today. That's two
I look
listening
much
one.
better than you do.
Oh, that's not nice. Oh, you're
Just
saying I don't have any hair? Do you see a bald spot here? Where?
I'm not saying that you don't have any hair. I'm not saying anything.
First, you don't respect my 40 years of experience, and now you don't respect my hair.
Don't be ridiculous. Of course, I respect your 4 years of experience. I called you this afternoon and we discussed something that I thought was wrong. Remember?
I do.
So, what do you mean I don't respect your 40 years of experience? I called the brain to consult it out.
Say they think you're a vibe. Okay, you are a vibe.
Fine,
I think that's
I'm a
fine. I think
vibe.
it's cool, even if it's not right. I like it actually. I like being a vibe.
Good.
I'm not a vibe, but you're a vibe. That's good. What things do
You can be a
SARS-CoV?
vibe.
I have a different vibe. What things do SARS-CoV-2 have left to teach us? Amy would argue that it hasn't taught us anything yet.
I'm still waiting.
But if someone asked me what important concepts have been learned, what principles have been learned from SARS-CoV-2, I would say none. No principles, no fundamental principles. Right, Amy? Would you agree with that?
Yeah, I would agree.
Okay.
with that.
But what do we have to learn? I think we need to learn how we transition from this state where people with poor memory, immune memory, are dying to a state where when you're infected, it doesn't matter. I think that's very interesting because that will tell us how the common cold coronas became what they are today, I think. But I think it's going to take a long time. I'm not going to be around.
I don't know that I would say it's going to tell us how the common cold coronas.
Yeah, I know, not
became
exactly.
No, what I think it tells you is how the immune system in general works. I don't think it's
Yeah,
pathogen
I think that's
specific.
fine. You know,
I
I
think
agree.
it's how the immune system works.
Yeah.
But I think what SARS-CoV-2 has not hasn't really taught us anything except that we're arrogant. We thought we knew everything, and we then didn't really remember what we knew, and then we misinterpreted and said things that were incorrect. And many of those things were fundamental principles. So we were arrogant in our thought that we understood all these fundamental principles. So we had to go relearn them. Like that antibody responses contract.
But not
You would
everyone
say that that's
felt that. Not everyone thought it was a defect, right? Some of us knew that it happened, right?
Right, but the loudmouths on Twitter thought it was a defect, right?
Yeah, I agree.
So and some of those loudmouths are very prominent immunologists.
That shocked me, totally shocked me. But I should have
So
known.
I think that they assumed that they knew something, were arrogant about it, and then in the end, they had to go back and relearn it.
So Nancy said, I think we learned that fear is a powerful emotion.
No, we didn't learn that. That's how all authoritarians come to power. From Alexander the Great to Napoleon to Hitler to Trump to Putin. That's how they all come to power.
Hey, folks, click the like button before we move on. It's nice to get over 300. We got 460 of you. Just push like. Why? Because it makes other people more aware of what we're doing. The more likes, the more likely that is to happen. And so we want people to come to us where they get good information. Amy, encephalitis is all in your head. It is, right?
Yeah.
Here in Japan, a soccer team announced 3 players who got infected. No symptoms yet, although one has a temperature of 36.9. These players could play and have no issues, right?
Sure.
Well,
I
here
mean,
they would.
I don't know if I would say they should play. They could play. I mean, would you play if you had a cold or a flu with these symptoms?
Well, they don't have any symptoms, so. But the
Okay,
problem is,
well, they have a little bit of an elevated temperature, right? I don't know.
it
36.8 is
really,
body
I
temperature.
I would think it's okay. But the problem is that what if one of the other players has some at-risk condition? You don't want them to be infected, right? So here they would
Why,
not
you
be
don't give
allowed
a
to
shit
play.
if you do that for flu?
Are you sure?
Hmm. Do you ever hear people say, I'm not playing because even though I have flu and I don't feel bad and I don't really have any symptoms, that I'm concerned that my opponent is immune compromised and is going to die of flu? No.
No, but there's a special place for COVID because people are afraid of it.
Well, so that's the problem. Don't make special rules.
Well, I agree, but I didn't make them.
Well, I don't think we should endorse it.
Okay, I won't endorse it.
Because still, how many thousands of people die of flu a year? And there's somebody's relative, there's somebody's sister, grandparent, kid, something or other.
A lot of people can die depending on the year.
Yeah.
Yep.
So,
Well, we've gotten used to that. We're going to get used to COVID, right?
right. So therefore, when people say it's not flu, you're just accepting the amount. It's okay that you die of flu, but that's not right.
So Doreen said she got
Death is death.
Paxlovid for just being 64, no other issues.
Okay. Well, every doctor interprets the EUA differently, I guess.
Well, you're not supposed to.
To
No,
tell
I
you.
will not replace Dr. Fauci because I'm not an MD. But if Amy were an MD, she could replace him. But she's not an MD, so neither one. I'm really curious to see who they will tap to replace him. Correct. The new RSV vaccine is being tested on over 60s. Yes.
Wasn't that a science paper?
The
Some of the
RSV vaccine?
results?
I don't know.
I
Maybe.
think I sent you the paper.
Okay. Yesterday I discovered that no envelope virus is spread by fecal oral route. Well, that's not correct, Liam, because there is a transmissible gastroenteritis virus of swine, I think, which is a coronavirus, which is enveloped and which is spread by the fecal oral route.
Porcine diarrhea virus.
Porcine diarrhea. So there are animal viruses that are enveloped that are spread. It wouldn't be an instant refutation to SARS-CoV-2 being present in feces because there are examples of that. Yeah. hard to repeat, but is antibody escape equal to worse infection? No. Worse infection is largely caused by a poor innate response. Anecdotally, B5 wave seems to be, this is not worse infection now. You get infected if you have B5 and you have ancestral vaccine because there's less neutralization, but you don't get severe disease. So, that's not the correlation. You get protected against severe disease. You may get infected. But worse infection isn't correct necessarily. Old people can die from the common cold. Right. That's what Amy was saying before, right? And Richard. Hello, Richard. Kiaha saponins are also used in beer for making foam. Those are the adjuvants.
Cool.
Kansas hospital are overwhelmed. Many reasons, but COVID has really broken open the weaknesses. Scary with ERs closing all over the country with not enough staff. That's just unfortunate. I'm sorry to hear that, but I have heard that.
I think it's happened here.
Yeah, it has in places. That's correct. I've read contradictory papers on whether Evusheld neutralizes BA5. It's, according to Daniel, I asked him last week, it does. And the contradictory paper, well, that's what happens when you use different methods, right, to do something. Yes. There's no standardized method for looking at that. But you're still likely protected against severe disease unless you're in those special risk groups, right?
Right.
Did I forward you the email? I did, right?
Yeah.
As a virion floats around hoping to latch on, well, there you're first wrong. It doesn't hope. Viruses don't hope. To latch onto an ACE2 receptor, how long does it remain infectious? Where are you floating around? In someone's respiratory tract? Remember, there are lots of things going on in the respiratory tract. Things are moving. Mucus is moving. So you're not going to just sit there on top of the cell forever. You're moving around. Maybe some macrophages are coming in. So I would say in your respiratory tract, you may have a few hours. Right, Amy? Are you tired?
A little bit.
All right. We're almost done.
No, it's fine. Yeah, I mean, there's tons of stuff that's happening. I mean, pH could change and various, there's tons of stuff happening. I haven't talked to my mucus tract lately, but yeah.
I saw a guy on the metro with monkeypox in Washington. Well, I don't know that you could know it was monkeypox, right?
Yeah, I don't know.
Someone asked that.
I think the pox, though, are very distinct. I think they have a very distinct marking to them. They don't look like measles or chickenpox. I think they are very distinct.
Would a previous molluscum contagiosum infection confer protection to other pox? Good question. I have to ask Condit that. Because that is a pox virus. Protect against monkeypox. But I'm not going to find it, and I'm going to ask Condit the next time I see him. Most common human disease resulting from pox virus infection. I don't know the answer. Good question. But as you say, there is a lot of cross-reactivity, so I would not be surprised if there is cross-protection.
Mm-hmm.
Christina said, you guys, my microbiology teacher told me about you, and I've been watching you every time. I go, well, good. Thank you, microbiology teacher.
Yeah.
Thank you very much. Oh, okay. So there were two guys, the spots on his arm, he was scratching them, plus he got the train at DuPont Circle. I think he was I'm not sure you could conclude all of that. Anyway. I also work in the pharmacy. It's crazy how doctors prescribe Paxlovid and not educating people. So now I understand that pharmacists can prescribe Paxlovid, according to Daniel.
Interesting.
Pablo says there's some virologists that say monkeypox is serious and it's scary, but the TWIV team says otherwise. As a virologist, I don't know which sources to trust. I don't know why you wouldn't trust us, but you don't have to. We tell you why we think it's not serious. I mean, as Amy said before, we should vaccinate. We should ring vaccinate because you don't want to take a chance. But right now, it's not a severe disease. There have been a few deaths out of all the cases. But what if the whole world got infected? Then there would be a lot of deaths. So serious and scary are subjective words that really don't belong in the scientific lexicon, right? What's the capacity of the virus to cause disease? That's what you should be answering. A scientist shouldn't say this is serious. Well, serious may be okay, but scary doesn't belong in there. And then you have to ask them.
You can't really determine what the capacity is of a virus to cause disease because that's influenced by the environment.
Well, I think
hugely
what I mean by that,
influenced by the environment.
Okay, what I mean by that is in the human infections we've seen, how severe is the disease? Okay, that's all. And by severe, how many people die? And not that many people have died. You know, you can argue that one death is too much, and I agree with that. But when in public health, you know, you have to take a broad view of things. And so I don't think at that level it's scary. I think it can be, if we decide to vaccinate, we can control it. We eradicated smallpox. We can control monkeypox. So it's not an issue.
Well, so the thing is, so I think that one of the concerns isn't just death. I think the other concern is the disease, right? And the disease, I understand, is extraordinarily painful.
Yes. It's kind of like smallpox, very painful. Yep. Is BA5 more contagious? It wasn't more. No, it's not more pathogenic. No, I haven't seen any convincing studies that say that. And I think it's over-sensationalized, just like this latest one, 1.25 or whatever, that's in India. It's being over-sensationalized. Just get vaccinated. You'll be fine. What are good educational virology books that you recommend? Well, it depends on what level, right? Are you leaving us, Dr. Rosenfeld?
No, I'm turning off the air conditioner. It's too cold.
Oh. What are good educational virology books? Well, you know.
If you're like very beginning, then the lady's book about the cartoon character. What is it? Peter or Paul? Paul gets vaccinated and Paul gets measles, right?
Well, that's for kids, right?
Who knows? Well, what is that? What age is this good educational virology book supposed to address? Is it supposed to address kids or it's supposed to address, college people?
So, Alec, give us an age that you'd like because otherwise we're flailing.
I don't understand.
BA 2.75, that's the number, yeah.
Okay, fine.
So does virus identification via metagenomic methods only work in rare situations?
No.
No, I think if you...
Works better if you're in a dominant situation because then you can find a lot of the DNA. When you're in rare situations and it's unknown, it's very hard to piece together. You could piece it together wrong.
But I think you can't identify it unless there's a hit in the database. So it has to be related to something, right?
Right. It's always dependent on how good your database is.
What are the causes of high mortality from monkeypox in Africa? Well, that's a good question. Some people think it's a different variant of the virus, but I'm not convinced that it's not, it's not healthcare effects and so forth. I think the jury is out. Any plans to have Karate on to discuss his recent report showing very low likelihood of immune suppression from COVID infection? He might. be on when we go, if and when we go to Hawaii.
Oh, he's going to Hawaii?
Well, he's going to be at the meeting, so we're considering having him, but I'm not sure.
Oh.
We are still wearing masks at the hospital and will be with most patients for a long time. Yeah, it's a high risk. I totally agree with that. Does the new OPV make you hopeful that polio can be eradicated? No, I think we don't have enough data yet. And I think the makers are not even sure of it either. One of the makers at least told me, who knows what's going to happen. What do you think of that, Dr. Rosenfeld?
I agree with that. So we already know from what some work it does revert.
Yeah.
And it picks up, it recombines and picks up stuff. And you don't know how long those recombinants are going to hang out and be around for. And you don't know if they're going to recombine or pass through and develop other things, because some of those recombinants have reversions that associate with virulent properties. Mm-hmm. I'm not surprised. I'm pretty happy that the person who told you was honest, because I think we spoke to his partner once and he was kind of didn't really want to take a stake at that. You know?
Yeah. If the risk of long COVID after vaccination is so indeterminate, why wouldn't it be reasonable to wear a mask until we have better data? Well, I think that's your decision. As someone said at this meeting, we're virologists. We know the risks. I don't know if we know them, but I think you have to make a decision based on the risk. And if you feel it's indeterminate, I would rather see data on the actual frequency. Until then, I'm going to behave the way I am. So, but I totally understand if others want to behave differently.
Well, it's just looking at the glass one or two ways. You could look at it as half empty or half full.
Yeah, I agree. Let's see what else is here in the last few minutes or so. Those who got long COVID, first, second, or third infection, or is it random? Let's see. What did Daniel say about that last week? I think it's less likely at subsequent infections. The first is the most likely. Is that correct? Yes?
Yes. Yes.
Richard said Beth is from the word house.
Oh.
Okay, there you go.
Guess so. I did not know. Thank you.
We have a few
Maybe
people.
It's from from Bet.
What's your feeling about Geert van den Bosch hypothesis? I don't think it has any merit whatsoever. Okay. He is wrong. You know, he thinks we shouldn't be vaccinating because it's going to drive variants, blah, blah, blah. It's ridiculous. It's just totally ridiculous. Kathy is a great dancer. Yeah, she's a good dancer. She's a good dancer. Did you and Amy read the editorial by Iwasaki which asserted we need boosters all the time? She compared COVID to influenza,
I did.
Which was not accurate. What do you think, Amy?
I think they shouldn't be publishing anything else by her.
I think it's wrong. I don't understand.
Well, yes, but she's written at least two that have been completely wrong and stuff. And she's co-authored another one that has been half wrong. So I think she should not be published anymore. But, what do I know?
Well, you can't censor people, so.
Well, I think that they actually may solicit opinions from her. And they should be assessed.
No, we're not going to need boosters all the time. At some point, we're going to figure out how to deal with it. And I don't care. I don't think your opinion should drive that kind of conclusion. You should look at the science. And if you do, the Peter Marks, who is the head of CBER, was quoted as, they asked him, what keeps you up at night? And he said the realization that we can't keep on boosting. All right, so that's the bottom line.
I know.
We can't keep on boosting. So for her to say we need it all the time is simply wrong. I'm sorry.
Well, I mean, the truth of the matter is he's also saying something else. So she's only focused on the immune response. He's saying something else that is directed towards the immune response and the platform.
Right.
So the platform everybody thinks it's this fabulous platform, but maybe it's not so fabulous after all. So, she's not taken any of that into account. But she also wrote an editorial piece saying how we needed to make a sterilizing vaccine and blah, blah, blah. And you show me any vaccine that's sterilizing.
Right.
So the woman who purports herself as the immunologist of immunologists might want to rethink her platform herself.
Could I ask a philosophical question? Statistics, good or bad for science?
Bad.
Oh, there's such polarity on that question, Seth. Really good question. We had discussion at ASV. I mean, some people really like statistics. Epidemiologists really like statistics, but
yeah, because they know they're basically mathematicians who know how to manipulate it so that you could say whatever you want, and they're good at using words. So again, they can make it say whatever they want.
But I prefer an experiment that doesn't require statistics to tell what the answer is. And I understand not all of them do, but some people are wedded to statistics, and they think that they tell you about your outcome, but you have to look at it biologically. If you want to use statistics, that's fine, but don't be, what do they call it? The tyranny of the p-value, right?
Yeah, the tyranny of the p-value, my tuchus.
Paxlovid was awesome, but the side effects, yes, people tell me that the taste side effects were horrible, right? But it makes
my
you feel
uncle
better.
is on Paxlovid, and he didn't like the side effect of the taste, and so he stopped.
Yeah.
And I was like, you shouldn't do that. And then I was quickly informed it was already done and I was not an MD. I was like, okay, fine. Whatever.
I remember before the discussion was spike was not the best answer. Well, I think spike alone has been mixed, right? It's saved a lot of lives, but it doesn't have all the T cell epitopes we would like. So more protein, as Amy said, if you had the whole genome in there, it would probably be better. But I don't think pushing it to do things wrong is correct. I think, natural infection would have
100% vaccination may push it to do things wrong.
No, that's
I
not
don't
correct.
understand that.
Well, she's saying I think the interpretation is, does it cause variants to emerge? And I don't think that's the case. They would emerge anyway in the face of immunity.
For sure.
When the individual is at higher risk of contracting severe disease despite vaccination, do we need to be clear that masking refers to masking with a properly fitting mask? Yes, but some people don't want a mask, and it's no longer mandated in certain situations. And so in that case, you should be ready with antivirals or monoclonal. Okay. I hear young people with no med issues getting Paxlovid. That's true, and they shouldn't be. It's not in the EUA. That's true. Reality is many doctors will not prescribe anything for COVID. This includes Paxlovid. That's true, and that's why some people are dying. Absolutely. Yep. I don't know why they aren't giving Paxlovid to everyone who can swallow. I have thought of this myself. Yes. Why not? Because even young people could die because you don't know if they have an underlying issue that you're not unaware of. So,
well, I also could make that argument for why more people don't get monoclonal therapy. I think monoclonal therapy is still restricted to only severe risk.
Yeah.
I don't know why. Put the stupid thing in a bioreactor and make a ton of it. It's not that big a deal.
Yep, I agree. I don't know why we have to restrict it. I think it could be
Not
really
at this point in this time. Not at this date in time. I mean, maybe 2.5 years ago, fine, but not this point in
yeah.
time.
All right, let's make our way through here and see if there's anyone else to thank. We got to thank all of you. Gina, thank you for your contribution. Very much appreciate your support of science education at the incubator.
Stay tuned for the polio video tomorrow.
I'm going to make a polio video explaining the case in light of science. And now that I've announced that I have to make it, right, Amy? I will do it tomorrow. I have some time because the immune has just been canceled, so I have until 3 p.m. to do it.
Why was happening at 3?
I have the Laura Sicola podcast.
Oh, yeah, I forgot.
Anyone hear of that? Laura Sicola, S-I-C-O. Well, I'm going to be on her podcast tomorrow.
I forgot. Got plenty of time.
Why do you need 2 swabs to collect pustules to get enough material? I think it's not abundant as you would like, so you need 2. It's my understanding. Anyone else seen the Times headline equivalent to Biden has COVID now what? And think, what do you mean? Now he gets treated as if he's sick and then he moves on. Why is it a question? To get people to read it, Caroline, right? That's what the press is all about. That's what the press is all about. Oh, I'm so sorry, Arnaud. Your house had a fire. I hope everything is okay. We missed you. I haven't seen you in a while. Nasal vaccine, haven't heard anything lately. Thank you, John, for the contribution. Thank you very much. Really appreciate it. So you saw some new faces here. That's cool. Thank you, Siddhartha, for your contribution to science communication. And that'll do it for another episode of Q&A with A&V. Thanks to the moderators. Let's see, we had Les, we had Steph, we had Vanity Nutrition, we had Frank. Only one missing was Tom, who is unavailable. Thanks all of you for coming. And I'm very grateful you could make it on a night that's unusual. Unfortunately, next week we're not going to have a Q&A, right, Amy? Because I'll be away on Wednesday and Thursday. I'll be in Colorado, Fort Collins, for a BAT meeting. So if anybody's in Fort Collins and you want to meet up for a drink, I'd love to. Just send me an email, vincent@microbe.tv. Meanwhile, bring your questions back in 2 weeks so everybody gets a break, including Amy. Amy needs a break, right? You need a break from Q&A?
I'm fine.
You're fine. But anyway, thanks for your questions. They're all great questions, and bring them back next time. Remember, in 2 weeks we'll be back because I'm away. And Amy, Dr. Rosenfeld, thank you so much as well. I appreciate your participation.
It was good. It's good. I'll see you in 2 weeks.
Night, everybody.
Talk to you tomorrow.